This meeting is being recorded.
Welcome. Welcome to the GSA webinar series on National Advisory Council, NIA Priority Research Concepts. This particular webinar is on Alzheimer's disease, research centers, and related resources. Next. Please use the chat box to introduce yourself, and you can also ask questions throughout the presentation. Our GSA staff will help resolve any technical issues you may have and also provide the questions to our presenters. Do know that this conversation is being recorded and will be archived along with slides from all the presenters on GSA's website in the near future. And if you are pleased with today's format, please let your friends know, and also let us know what you would like for future topics on future webinar programs. Next slide. I am James Powers, the lead for the work group that developed these webinar series. My colleagues are Daniel Gann, Sam Lee, Meredith Troutman-Jordan, and Pan Satoris, all members of the Gerontologic Society of America. Next slide, please. It's my pleasure to introduce my colleague, Stephen Krichevsky from Wake Forest University, who will serve as our moderator and introduce our presenters on Alzheimer's disease, research centers, and related resources. Stephen?
Thank you so much, Jim. I'm delighted to serve in this role today. Alzheimer's programs are incredibly impactful, and I think we all will benefit from hearing more about those opportunities. To kick us off, we're very pleased to have Dr. Nina Silverberg, who is the director of the Alzheimer's Disease Centers programs in the Division of Neuroscience at NIA. And she will be starting her slides, and I will turn it over to her. And you are also muted, Nina, so you may wish to unmute.
Okay, I'm unmuting, and I'm about to share my screen. Let's see if this works.
Perfect.
Great. Okay, thank you for inviting us to share information with the GSA about the Alzheimer's Disease Research Centers program. I am the director of the program along with my co-director, Cerise Elliott at NIA, and we have assistance from Grayson Donnelly and Alexandra Dakic, who just joined us recently. Let's see. And so I'm going to share a little bit of background about the Alzheimer's centers themselves and share with you the general overview of the infrastructure. And then you'll be hearing from the rest of the team today about much more detail about the different pieces of the infrastructure. So we, in 2019, funded one new Alzheimer's center in Ohio. You can see that one. In 2020, we had a unique RFA that was for P20 exploratory centers, and our goal with those was to increase diversity along several dimensions. One was geographic, which you can see we succeeded in. One was scientifically to get new and interesting, innovative approaches. And the last was populations. And we do have, as I'll be showing, considerable diversity in the different populations at each center, but we really had a goal to massively increase the diversity by including these new P20 centers. And their goal is to develop the center enough to apply for a full P30 Alzheimer's Disease Research Center. And so in 2021, we actually added 2 more sites. There's a star in North Carolina over here because we already had one center there. And now in 2021, we funded a new center that's actually a collaboration across two physical institutions, UNC and Duke. And then in Texas, the same thing is also a collaboration across two different centers, one right at the border in Brownsville and one in San Antonio led by Sudhis Seshadri. And the one in Brownsville is led by Gladys Maestra. So that's our current map is 33 Alzheimer's centers plus 4 P20 exploratory centers. And we have a new RFA that just came out very recently, and that's why I guess we got flagged for this presentation. It's RFA AG 24001, and it has lots of new and exciting things for the centers program that we hope everyone will take a look at. Even if you're not applying to be a center, you can see the types of resources that are available that I'll be talking about now. So here's, as I mentioned, different types of diversity represented across the Alzheimer's Centers program. And please note, these are examples. They're definitely not the full array for each place, but just filled in some ideas of the types of underrepresented groups we have at various centers across the country. And then some specific populations that individual centers focus on: Down syndrome, DLB, FTLD, oldest old, rapid progressors, and veterans. Those are just some examples of the types of populations we have across the center's program. So this is the slide I'm going to spend the most time on to help folks understand what we have available across the centers. So the overarching plan is, as I mentioned, we have 33 ADRCs plus 4 exploratory ADRCs, and these cover 26 states. Each of the Alzheimer's centers collects a uniform data set, and all of that data is sent to the National Alzheimer's Coordinating Center in Seattle. Each of the centers also collects brain tissue from autopsy that they keep locally, but also shares some samples to NCRAD, the National Centralized Repository for Alzheimer's Disease and Related Dementias, which is in Indianapolis in Indiana. NCRAD then shares the samples out to other investigators and also shares biomarker results back to NACC, the coordinating center, so that all of this information is available to outside investigators. NACC data is also shared out through the Global Alzheimer's Association Interactive Network. And so this is another way that we make that data available. We've recently also funded SCAN, which is the Standardized and Centralized AD Neuroimaging resource. And that is a collaboration across, again, multiple places, but primarily led in Berkeley by Bill Jagist. And through this, prospectively, the Alzheimer's centers are collecting MRI and PET data, amyloid and tau PET, in a standardized way, which is the new piece. And that standardized data is analyzed, and then the data is sent also back to NACC for further sharing. And then we have the DNA and biosamples that get genotyped and sequenced, and you'll hear a lot more about the ADGC, Alzheimer's Disease Genetics Consortium, the Alzheimer's Disease Sequencing Project, and NIAGADS, where genetic data is stored in a database. And these are all interconnected and made available to researchers. So I hope, despite all the crazy arrows going every way, that that's somewhat clear what's available where. And here's a little bit more information about the uniform data set, which actually we're just in the process. This is the uniform data set version 3. We're just in the process of developing version 4. But it collects demographic information, co-participant information, family history, medications, a full cognitive battery, a clinical diagnosis for research purposes. And these are all incorporated into many, many national projects and utilized for various types of research. So you can see that this uniform data set is really helpful for the whole field, we hope. And here's just a little bit more information about SCAN. You'll be hearing a little bit more, but basically the ADRCs collect the data, as I mentioned. It's made available through NACC and LONI, and then those slides are made available, the scans are made available. And this is my last slide. I'm just sharing the infrastructure. Each Alzheimer's center is required to have 6 cores plus a research education component. And then some of them also have optional cores, maybe for specific populations or methodologies, like some have IPSC cores, for example. So they all have an administrative core, a neuropathology core. All of them do autopsy and send autopsy data that's also stored at NACC. Each of them has a clinical core with research participants who are seen every year, and then an outreach, recruitment, and engagement core that interacts with the community and helps with recruitment and retention, a data core that sends all the data, and now a relatively new requirement for a biomarker core, which can be fluid, imaging, or even digital, or any combination of those. And then this research education component. So I'm going to stop sharing and let Dr. Krzyzewski introduce the next speaker.
Thank you for that wonderful overview, Nina. It really helps set the frame for the rest of the presentations. The next speaker is Dr. Sarah Biber, who's Program Director at the National Alzheimer's Coordinating Center. Go ahead.
Here and unmute.
We can hear you.
Okay, can you hear me all right?
You hear it?
Great.
Slides look good. Voice sounds good.
Excellent. Hello, everyone. Yes, I'm Sarah Biber, and I'm the Program Director at the National Alzheimer's Coordinating Center, which is directed by Bud Kukul. And I'm fortunate to have the opportunity to work closely with some of the other folks you're hearing from today, including Nina Silverberg, Tatiana Faroud, and Lisan Wong. Sorry, my arrows. NACC serves as a data collaboration and communication hub for NIA's Alzheimer's Disease Research Center program. As Nina mentioned, there are currently 33 ADRCs and 4 exploratory centers that are spread across 26 different states. And today I'm going to use my time to tell you more about Alzheimer's data that we collect, integrate, and harmonize from across the ADRC program, and how we are sharing this data with investigators around the world to help advance Alzheimer's research. So here's how it works. There are 37 independent centers around the country that collect standardized longitudinal data. Centers enroll research participants who are healthy, at risk, or with dementia symptoms. And centers then collect that standardized longitudinal data on those participants through these annual visits using tools that are co-developed and provided by NACC. So NACC has been doing this for a while, for more than 23 years, and we've been tracking some participants for up to 18 years. Over these past 2 decades, NACC has been focused on collecting, harmonizing, integrating, and sharing this ADRC data. So we have more than 47,000 participants in our data set to date. That includes more than 16,000 active participants. There's data from more than 174,000 clinical assessments. We collect a lot of neuropathology data as well. We currently have more than 7,000 neuropathology data sets. This represents 58% of the deceased participant population. And there are currently more than 1,000 published studies that have used NACC data. So the NACC data set is uniquely valuable. It represents one of the largest and most comprehensive longitudinal standardized clinical and neuropathological data sets in the world. It includes participants with normal cognition, MCI, and dementia, as I mentioned. And for many participants in our database, data has been captured prior to and post-dementia onset. We have also have the infrastructure in place, as I mentioned, to track these participants well into the future. We have robust criteria-based diagnoses. So these are often referred to as kind of gold standard diagnoses. At many centers, a consensus diagnosis must be reached by multiple specialists. The NACC data set also consists of incredibly rich multi-domain neurocognitive data. I'm going to tell you some more details on exactly how rich this data is shortly. In addition, as I mentioned, we have neuropathology data, and this includes for diverse etiologies that are all represented within our data set. And the rich phenotypic data that we collect has been a really valuable tool for genomic analyses. I also want to mention that NACC, I mentioned that NACC already collects and integrates multimodal data. So this currently includes standardized clinical and neuropathology data, as well as non-standard MRI and PET data. And collecting this multimodal data across a large scale observational cohort such as ours has been valuable for really powering data-driven research examining heterogeneity in ADRD. And these data have already allowed for a richer understanding of the variability of disease expression within aging and the ADRD continuum. So I'm happy to report that NACC is in the process of becoming much more multimodal. So we are currently launching a new modern data platform that will allow for the integration of new standardized data streams. These data streams will include digital biomarker data, digital pathology data, as well as EHR and CMS data. We're collaborating closely with NIAGADS and NCRAD to integrate more genetic and biospecimen metadata from ADRC participants, as well as with SCAN initiative to integrate standardized MRI and PET metadata. So we're really excited about the integration of these additional standardized data streams, and we're hopeful this will allow for more differentiated disease stratification and will be important for improving individualized diagnosis and prognosis of expected clinical progression. We're also optimistic that this new resource will create more opportunities for the development of precision medicine approaches and for therapeutic intervention. So in summary, the unique features of NACC data really make it viable for research across the ADRD continuum, including studies of early to late ADRD onset, risk factors and progression, studies focused on disentangling and discovering new etiologies, as well as studies aimed at developing machine learning tools to facilitate dementia diagnosis and the differentiation of underlying etiologies. So now I'm going to briefly share a few more details about the data we currently have available, as well as what is coming online soon. So our more than 47,000 participants include individuals of different ages, genders, educational levels, races, and ethnicities. As Nina mentioned, there's work happening right now to diversify this data participant pool even further. As I mentioned, the neurocognitive, clinical, and phenotypic data that we collect is incredibly rich. So this includes detailed health demographics and family history, as well as detailed neurological exams and caregiver interviews are conducted to assess participants' cognitive, behavioral, and motor condition. Additionally, there's a comprehensive neurocognitive battery that is performed, which includes the MOCA, as well as 12 additional tests to assess language, attention, executive function, and memory. In our new version of UDS that Nina mentioned, which we're calling UDS v4, we're also in the process we're in the process of launching right now in collaboration with the ADRC Clinical Task Force. We will also be starting to collect social determinants of health data, which will really open the door to studying new and important aspects of ADRD. As I mentioned, we have a wide range of ADRD etiological diversity represented within our data set that is shown here in terms of how much of each of those varieties we have. This was all data that was, this is all assessments that were made using neurological exams and testing. In addition, these diverse etiologies are also represented within our neuropathology data set. So this slide shows some of the pathologies that you can explore using NACC data. In the future, we're hoping to also be able to integrate digital neuropath data into our data set, as I mentioned. So I want to say a little bit about SCAN. So, oh, sorry, this is the wrong slide. Things got out of order, apologies. So NACC already integrates some genetic and biospecimen data and metadata from across the ADRC program into the NACC repository. As I mentioned, this is done in collaboration with NCRAD and NIAGADS, and we hope to be doing more of this kind of integration in the future. We have APOE data for 73% of our participants. There's GWAS data available for more than 16% of NACC participants through NIAGADS. In addition, we have some CSF and blood biomarker data that is available for NACC participants through NCRAD. And in the future, we'll be working to integrate metadata for top genetic variants, as well as additional standardized blood biomarkers. So a little bit about SCAN. So NACC is also in the process of working to collect and integrate standardized MRI and PET data in collaboration with the SCAN initiative. So we already have within our database some non-standard longitudinal MRI and PET data available. You can see about 10,000 MRI images, about 500 PET images. But coming soon, literally like in the next couple of months, there's also going to be more and more standard MRI and PET data that's available. So that data is collected from across the ADRC program. It is stored at LONI and will be shared through our NACC portals. And that includes analysis data like including volumes and PET SUVRs. So this is, we have how many were available as of last month. And as I mentioned, there'll be more and more of that becoming available soon. So NACC data is being used for impactful research all over the world already. And I want to just share that if you don't know, it's freely available to all researchers. It's very easy to get. This is about I can submit a request in about 15 minutes. This involves signing a data use agreement, submitting a title with specific aims. The NACC team then reviews this request, and requests are typically approved and data is delivered within 48 hours. NACC also provides some support for researchers. So this includes guidance documents as well as analysis support documentation. We also provide some in-depth biostatistics support for some select collaborations. So you can learn more by going to naccdata.org. These slides will also be shared later on. And with that, I'd like to thank the amazing team at NACC as well as the NIA for providing funding and support for this work. Thank you.
Thank you so much, Sarah. Excellent overview. If you have questions for the panel, please put them in the chat and we'll address questions of general interest after all the presenters have spoken. If there's a very specific question, you can direct that to the speaker. If it's but if you think everyone would enjoy hearing the answer to it, please put it in the chat. Our next speaker is Dr. Tatiana Farud, who is the principal investigator of NCRAD, because people don't want to say National Centralized Repository for Alzheimer's Disease and Related Dementias, because their time is up when they're finished saying that. So take it away, Dr. Farud.
You've stolen my joke that I always open with.
Sorry, so sorry.
So delighted to be here, and I'll try to take you through a similar tour of what we have available for researchers. So let me start by saying I think our scope is relatively easy to describe. We take samples in, and our goal is to get samples back out again. So what I'll try to walk you through is the diversity of samples that we have so that you can see how we can support your research. So we bank samples from 65 different studies, and I'll give you a little bit of a flavor in a moment. This encompasses over 100,000 individuals, unique individuals that have samples with us, well over 1 million aliquots of samples. And we've distributed to 249 different researchers. I'm looking for the 250th. I would love that to be you. And that's nearly 400,000 sample aliquots that we've distributed. I think a good way to get a sense of the value of the samples is there have been over 700 publications using samples from NCRAD. And in a couple different slides, I'll always have our website down there so that whether you're on now or you're listening to a recording, you can see how to learn more information. So I described it as a range and a diversity of different areas that we support. We began really focusing on Alzheimer's disease. So early onset, late onset Alzheimer's disease, and then individuals at increased risk for Alzheimer's disease. We've had now a decades-long collaboration with the Alzheimer's disease research centers. Increasingly, there's been a focus on studies of aging, whether it's healthy aging or individuals who are progressing potentially toward dementia. As there's been a growing focus on ADRD, you've seen that we have focused with frontotemporal dementia, Parkinson's disease and RBD, Lewy body dementia, as well as Down syndrome. Down syndrome, not so much sitting in the RD, but because individuals with Down syndrome have increased risk for Alzheimer's disease. So we really try to encompass the mission of NIA, which has grown to really encompass ADRD. This is a busy slide. I'll just try to give you highlights, but for those of you who look at this later on, hoping that you'll be able to spend a little bit of time looking at this. I've described to you sort of some basics about the sample banking. What I haven't described to you is the range of samples. So we do a variety of samples from blood. We have CSF, brain tissue, cell lines, iPSCs, and fibroblasts, increasing interest around stool. We make sure that these are collected in a uniform way. So if you go now to the top right, you see that we try to do all collections in a uniform way. We do that through a study manual of procedures that ensures sites know how to collect samples in a uniform fashion. We spend a lot of time on QC, so a variety of QC measures, so investigators receiving samples know about the quality of those samples. We track all freeze-thaw cycles. And as I'll show you in a little bit later, we also have online catalogs, and we have many, many aliquots. Our goal here is to support researchers. We want to make sure that researchers understand what they're receiving from us and how we can best serve their needs. So we do that by having a call with researchers who've been approved to receive samples. We have the ability to include reference samples. Those allow to control for batch effects and also to compare samples across studies. We'll place the samples to minimize how much the person receiving them needs to do. And we use blinded sample distributions. That way, an individual returns results and then is able to link them to the clinical data. That's to really help with the rigor and reproducibility that's become so important in our research. And again, we ensure that the data generated from samples are then posted and made available to the broader community through NIAGADS, NACC, and LONI. And as I mentioned, over 700 publications have been generated using samples from NCRAD. So to give you a feel, I've mentioned about the fact that we really focus on having samples be collected in a uniform fashion. On our website, you can see that we have a whole variety of services, again, ensuring that samples are collected in a uniform way to support studies. We also have a video that you can watch if you want to better understand what can we do in order to ensure that uniformity. How can you work with us? And to give you a sense, in order to be able to provide all the tools for studies, we have a kit room that ensures that we can track what's been sent out, ensure that studies are using the right tubes. We provide everything that they would need. We again know that people learn about things in different ways. So we've tried to host, just like here, we've also hosted a series of webinars on different topics so people can better understand the capabilities that we have. We have a webinar all around iPSCs and catalogs. I get a more detailed one just looking at our catalogs, and also one looking at outgoing samples. If you're interested in learning a little bit more, all of these webinars are available on our website, and really hope that that's a great way for you at your leisure to learn more about the way we can support research. Wanted to give you an example of what it might look like, and also for you to understand how we're all connected. And by us, I mean NCRAD, NIAGADS, and NACC. How do we all work together? We've all been really highlighting that we see ourselves as a group. We're like a three-legged stool, as Bud Kuka would say. So I'm highlighting this with the ADRC catalog. So this is a catalog that focuses, again, on the samples that have been collected at the Alzheimer's centers. You can see a whole range here, things like peripheral blood mononuclear cells, plasma, serum, DNA. If a person comes to our catalog, they can look at which visit from an individual, and you can pair that with clinical data. If you want to have samples paired with a particular phase or stage of disease, you can identify which visit you'd like to have samples from. We have the amounts that we distributed. We also describe how many aliquots we have. You can use this resource to filter, select samples. We try to make it very interactive so you can see what we have, and importantly, what data has already been generated from these samples. So you can see links to NIAGADS that shows that there's samples for these particular subjects already with whole genome sequencing, with GWAS. You can then go and also gather that data. So we try to make it easy for you to see how rich the data set is that's available on different individuals, and use that to get just the right sample that you need from us. We also have a growing collection of iPSCs, induced pluripotent stem cells. We've tried to make it easier for people to think about how to use them. We're starting to think about them as sets. So they may be particular lines that we have with certain mutations. Again, in certain genes, it might be particular ApoE variants. We know there's a lot of interest in being able to look at that. Variants, again, with known mutations. And increasingly, as people want to focus on functional studies, they want to be able to get iPSCs from individuals that might have been used in discovery that are part of existing studies. So individuals and lines derived from those individuals from the ADRCs, from LEADS, an early onset study, ADNI, which is going to be coming, All FTD Act. So we're increasingly trying to create resources that allow people to take initial results, work in the same study, and be able to follow up with them using functional approaches. We also now have a new capability around biomarkers. So again, thinking about our ATN framework, really laid down in terms of pathology, amyloid plaques, we've got tau, we've got neurodegeneration. We can look at that in the context of neuroimaging, but importantly, we now also have capability in terms of being able to also pair with ATN plasma biomarkers. And so we have new capabilities around that. We have both a Quanterix, on which we're able to do 4-plex, 2-plex. We also are able to run p-tau and NFL individually. Excuse me, and we've just developed capability using LumiPulse from Fuji Rubio to also be able to do A-beta 40 and A-beta 42. We see that as a really important improvement. There's much smaller variation in this assay, and we're just now also getting up their new assay around tau 181. So really pleased to be able to offer different platforms for biomarkers and hope that that's something that's also of interest to all of you. And that's where I'm going to end. I'm glad to take questions at the end of this session and hope that you'll put some in, and really grateful to have the opportunity to speak to you. Thanks so much. I think I'm turning it over to Lisa.
Sorry, I've been pretty good about unmuting myself appropriately. Our last presenter is Dr. Lisan Wang from University of Pennsylvania, who is co-directs the Penn Neurodegeneration Genomics Center. Take it away. And
All right, thank you.
Yeah, you got it.
Let me share the screen. Thank you so much for having me, and I appreciate this opportunity to share with you the resources, especially on genetics and genomics of Alzheimer's that we've been working on for the past 10 years. And I'm PI of the NIAGADS, which stands for National Aging Genetics of Alzheimer's Disease Data Storage Site, and we focus on genetics and genomics. The mission of NIAGADS is to set up and maintain a continuing data resource for human genetics and genomics research for Alzheimer's disease. And here's the URL to our website, which has a lot of information. We have more than 90 different data sets. That comes from more than 144,000 samples, and many of them come from the ADRC program. And in addition to your typical genome-wide SNP array data, as well as we have whole genome, whole exome data, and other kind of functional omics and proteomics data, analysis results, different kind of data. It's a really rich resource. Again, I invite you to visit our website and send us questions if you have any. And a lot of these data are based on this important concept, genome-wide association studies, where we collect the DNA from patients as well as copy normal age-matched controls, and then we compare them across the genome to see if there's any location that's associated with the risk of Alzheimer's. And that means there's something really important biologically in that region that helps us find new genes for Alzheimer's, improve our understanding about the biology of the disease, and some of them might turn into targets for therapeutic discovery. And one important thing about genome-wide association is you need really big sample size, and working with that's where all these infrastructure that was mentioned earlier play a very important role. And here's one example: Alzheimer's Disease Genetics Consortium, ADGC, that has been in place for more than a decade, directed by colleague Jerry Schellenberg. We've been working with the ADRCs because they have maintaining these very well characterized cohorts, and NACC and NCRAD already have the samples and the phenotypes, and they play very important roles to work with the ADRCs and help them solve the problems and encourage them to work with ADGC. So we have the genetic data, we have the phenotypes, we conduct genome-wide association, SNP array genotyping, together with some studies that focus on Alzheimer's, and we're able to assemble a really large resource with big sample size that gives us enough statistical power for gene discovery. And many of these findings are published in various papers, including some of the recent Nature Genetics papers. The latest one was last year, including international collaborations that led to more than 70 genome-wide significant loci associated with Alzheimer's risk. And we have some resources to present these findings. For example, Alzheimer's disease genomics database. We just released our latest version with using the latest human genome reference build, build 38. It has more than 150 variants from the various studies. Also, it generates detailed reports for the genes or the variants that you're interested in. You can go to the website and type in the records, and you can also view them on a genome browser that display all these gene and variant annotations and connect to different kind of functional significance and knowledge about these variants. So you can go to this URL below to give it a try. It's a fully featured genomic database. You can also visit this is another resource. It's a human curated summary of top findings in genome-wide associated studies. And the first release, which we published last year, has records from more than 120 studies published by ADRC, and has more than 7,000 association records. And you can find additional valuable information, such as the cohorts used for the analysis and so on. And it links to the genomics database and other resources. The other thing I want to talk about is the Alzheimer's Disease Sequencing Project, or ADSP. It is a National Institute of Aging strategic initiative where we study the Alzheimer's patients' genomes by conducting whole genome and whole exome sequences. So the goal is to find especially rare variants that have strong effects on the Alzheimer's risk that you cannot discover using your typical sniffer rate means. And that gives us insight about the disease, but also, of course, potential targets for drug discovery and therapeutic approaches. And the program is in its 10th year now, and it has continued to grow and have many additional new programs on functional genomics, AI machine learning, and clinical phenotype harmonization. And we have a lot of collaborators to come through this project. It's going to be a very valuable resource for the entire research community. We have a website dedicated to this project. It just released last year. It has a lot of information. I invite you to have a look. The latest release we have for ADSP and NIAGADS is release 4, which includes the joint calls all the genotype for more than 36,000 complete genomes of Alzheimer's patients as well as cognitive normal controls. And they have high quality whole genome deep sequencing data, and it covers more than almost 400 million variants across the genome. One thing I want to highlight is if you look at the composition of the samples, more than half are from underrepresented minority. And we're going to continue to do this because it increases the diversity of our samples, but also increase statistical power for gene discovery. The other thing that's very important is the addition of a phenotype harmonization consortium. And they work with ADSP, NIAGADS provides support for data management. And these are driven by domain experts in the various types of clinical data to collect data from the various studies, including many from the ADRC from NACC, and devise statistical harmonization approaches to put all these things together so these data can be analyzed together. And the first release we just released to the research community last year includes cognition, biomarker, and neuropathology harmonized from 8 cohorts. And we're going to have another major release this year that is going to cover more cohorts as well as more domains of data. ADSP also has a new initiative that just started 2 years ago. It's called Follow-up Sequencing Diversity Initiative. Statistical power analysis calls for 18,500 cases and the same number of controls to have enough statistical power for rare variant discovery in each of the 3 non-Hispanic, like underrepresented minority ancestries that we want to focus. So that will lead to up to 150,000 genomes by in 5 years. It's a very ambitious goal. So we're working with the not just US investigators, but researchers around the world to increase the sample size. And you can go to the ADSP website, as well as here's the RFA that tell you more about the details of the program. And ADSP is going to continue to update the progress of this sequencing and cohort expansion. Now, again, we just released our new website last year, and it has a lot of information about this important resource. And that includes the projects funded by NIA that are part of ADSP, a quick data guide, the procedures on how to request the data, list of publications, and list of a summary of the cohorts and sequence progress. One other thing I invite everybody to look at is we have a dedicated page on the top gene list that is reviewed by the Gene Verification Committee of ADSP. So you can think of it as high confidence findings from genetics that we encourage, we think people can focus on, because we have strong confidence that these are highly reproducible results from genetic study. And finally, all these resources, they are available on our data sharing service website, and it has detailed instructions and step-by-step guidelines on how to submit your data request. This is similar to how NI shares data through dbGaP. You can go to ADSP website, has more information as well. Just go to the DSS. And if you have any questions, if you're interested to get access to this data, feel free to just send us an email. We'd like to help you get access to the data. So that's my presentation. Again, this won't be possible without all the collaboration, all the help from participants, in particular the ADRC program. NACC and NCRAD are absolutely essential, and I would like to thank NIA for the vision and leadership. Thank you, and be glad to take questions.
Well, that was wonderful, and thank you, Dr. Wang, and thank all the presenters for just really crystal and clear, I think, very summoning presentations. Now is the time to ask questions. If you have them, again, put them in the chat, and we'll review them. There is one. I'm going to deal with a few more mundane ones because the question that came up is a good one, but can be pretty expansive. One question for Dr. Biber. Is there outcomes available, like hospital diagnoses or mortality associated with the records that are in the NACC data set?
Yeah, there is mortality data. So we have information on, who has passed away. And there's also, as I mentioned, neuropathology data that's available for about 58% of those participants that have passed away.
And for Dr. Farood, can anyone send you samples to have your panel run, or do you have to be an enrollee and have your samples at the NCRAD?
So I'm just looking because I didn't see that question. My apologies.
No, I'm asking these
So
on the fly. So
yeah, totally fine. So let me clarify. So I think you're talking about the biomarker laboratory.
I am.
Yep.
Yes.
So there's actually, so that was a great question. So the biomarker laboratory provides support for researchers that are banking with us, but can also run biomarkers for researchers that are not banking with us that just need to have biomarkers run. So we've really tried to make that as open as possible. The key thing is the mechanism by which, if I may say, that's paid for. So within the Alzheimer's disease centers, this is provided to them. So if an Alzheimer's center sends us a plasma sample, this is something we do and there's no charge for them. We can also partner with studies or individual researchers that are interested in being able to obtain biomarkers. Just has a different, obviously, cost structure because there's certain things that are supported within the NCRAD grant that is done in order to support that collaborations. Does that answer your question?
Yeah, great.
Perfect.
You. So we had one of the questions in the chat, and Dr. S, I'm going to say, because my Polish is a little rusty. About linkage between Alzheimer's and other neurodegenerative disease. But I want to ask this. I want to add on to that, and this is for Dr. Wang a little bit. So neurofilament light is one of the biomarkers of general neurodegeneration, And one would imagine that the expression of Alzheimer's disease is a combination of general neurologic frailty or susceptibility plus Alzheimer's specific factors. What about the genetics of the general, I guess, vulnerability to neurodegeneration that might also be evident in other kinds of neurodegenerative diseases?
Probably going to want all of us to answer this.
Yeah,
Go
that's
ahead,
fine.
Lisa.
Anyone who
You go
wants
first,
to-
but I'm guessing this is going to turn into a round robin, and we're all
Yeah,
gonna
that's
add
fine.
something to it.
That's
Go ahead,
fine. It's a
Lisa.
great question.
Yeah,
I thank the person for putting it forward, and it's really summoning. Thank you. Yep.
Definitely. So it is a complicated problem. Actually, starting off genetics make it a little bit simpler. So we do have some, like we have the progressive supranuclear palsy whole genome as part of the ADSP data. And of course, there are other similar initiatives, but for NIA, of course, like Alzheimer's disease is what NIA covers, and the others are covered, like say NIA is covered by Parkinson's. And Tatiana, you can probably comment more about Parkinson's. You're a leader in that area. We'd be glad to like work with other initiatives to get the data together. And the other thing about like, for example, we're thinking about mixed pathology. Like we've been focusing on how to make sure the data is like as homogeneous as possible in terms of the Alzheimer's. So when we find genetic findings, we know it's Alzheimer's, not because it's associated with some other diseases. That's why the diagnosis include like enrollment criterion is really, really important. A lot of studies we have, they try to make it sure it's mainly Alzheimer's, not other pathology. But I think we're starting to think about in that direction. I think that we're going to see more data down the road. And certainly if there's genetics, we can combine them together and do joint analysis.
So I'll add a flavor and then we can go around the table. So I absolutely agree. So there's also a lot of comorbidity. So individuals with Alzheimer's disease and individuals who have Parkinson's disease as individuals age, those are the two most common neurodegenerative disorders. And so it is not unusual to have individuals with both. So to sort of layer on that, what's critical is you alluded first to biomarkers. So that's why you don't just want to use, for example, NFL. That's why layering that and having a panel of biomarkers, tau, A-beta 40, A-beta 42, lets you start to make those distinctions. Of course, imaging, brain imaging can do similar things. So it's incredibly important when working with the populations that we are to recognize that we want to be able to identify and distinguish which features individuals have, which diagnoses, but also recognize that we may see both. I would just layer one last thing, and then I'll be quiet, is this is part of the reason why we generate ApoE genotype, not only in our Alzheimer's work, but also in our more broader neurodegeneration work. So when we work with Parkinson's groups, they also want ApoE because you want to be able to disentangle cognitive impairment due to Alzheimer's disease, due to other factors. So just, I think it's incredibly important. And the person who asked this question is spot on. And I'll let the next person tell them why it's a great question.
Yeah, if I could go next, just to add right on to that, in the new RFA for the Alzheimer's centers that I mentioned, we recognize, as the field has, that mixed pathology is the most common, in fact. And so our approach in the past had been, as Dr. Wang said, to kind of get a homogeneous group of people, and even trying to do that, we recognize that we haven't really been able to. It's just we've learned much more about how the real world works. And so trying to recognize that, we're actually encouraging a focus specifically on better understanding the different pathologies in the context of each other. And the new description of late, which is TDP-43 proteinopathy that occurs more often in much older adults in their 80s, is something that came out of the fact that the Alzheimer's centers have autopsy, and working with an international group that came up. And so I think this is critically important to be aware that it's very hard to actually find such a homogeneous group. And I just wanted to also mention that our NIA intramural program I'm putting in the chat, called CARD, although it's labeled with Alzheimer's, is also working in this area. And one last note is that the National Alzheimer's Project Act, which was signed into law in 2011, it was called the Alzheimer's Project Act, but it defined Alzheimer's as Alzheimer's and specific related dementias, which includes Parkinson's dementia, but not Parkinson's disease without dementia. So it's quite specific. But that has enabled a huge amount of new research that we do, NIA in combination with NINDS.
Sarah, do you have something?
Yeah, I guess, I mean, the thing I can add
add.
is that, I mean, this is why we're working really hard at NACC to make this data as multimodal as possible, right? So that you really have more data to, more data integrated to be able to differentiate between these mixed etiologies, what is the individual cocktail of conditions that is occurring in an individual's brain. And so the more, we're really excited about the standardized imaging data that we're going to be integrating, this MRI and PET data. I think that's going to be huge, being able to integrate these additional, this initial genetic data and this additional biospecimen data is going to be huge. Having all that connected to, for an individual participant is going to be huge. So just hopeful. There's already late was discovered, like as Nina mentioned, right? Using this neuropathology data available through NACC is a huge piece of that. And we're really hopeful we can both discover new etiologies with this data, as well as understand, better understand how to differentiate the mix of etiologies in the individual's brain.
Yeah, one other note on that, if you don't mind, and maybe this is about what Lisa was about to say, but is our effort to diversify not just the patient population, but the ethnic and racial and cultural background of the people who are participating, so that we have a much better understanding of the risk factors for these different forms of proteinopathy that can occur as people age, is really, really important. And so that's why there's such a heavy emphasis on that aspect as well.
Did you want to add something?
Yes. Following what Dr. Silverberg was talking about, diversity of samples gives a lot of research opportunities. One example is looking at the gene-environment interaction. Like for example, one of the projects that many of the people here are involved on, we're also expanding samples into Asian ancestry. That includes Asian Americans as well as Asians in Asia, which you'll see a perfect natural experiment where the genetics backgrounds similar, not exactly the same, but similar, but they have very different lifestyles. So I think that's a good opportunity to look at. The other thing I want to go back to that, look at different neurodegenerative diseases. That's one of the missions of NIAGADS and ADSP, but I think also is the principle behind NACC and NCRAD is that we want these data to be integrated, harmonized, and people can analyze these together. We know that studying these related dementia is going to give us insight of Alzheimer's disease, so we're going to continue to work on that.
So I think across all the speakers, you present this very, very comprehensive view of resources. But it's also, to somebody probably not in the game, a little intimidating. So what would your advice be to somebody who has a great interest or maybe even some training in Alzheimer's disease research, and they're not at an institution, they're not tightly connected to an ADRC? Do you have advice for them at all about how to get in the game? Or because I think we need as many people studying Alzheimer's disease as we can get. This is what I think. So Sarah looks unmuted, so you get
okay. Yeah, I guess I would say that we are actually in the process of revamping our website too, to make it really clear to folks that are new to the game, like how to use and access this data, what's available. But there's already a lot of information on our website about that, nacdata.org. And, we are happy to provide consultations with members of our research team too, to learn more about, ways to use this data. And I think another big piece of this, we are looking for create new on-ramps for other researchers. So one thing that NACC is thinking about doing is running dream challenges. And so as we create this new data platform that really is ready for like things like AI-driven discovery, right? We want to be able to make that data available to data scientists and other types of researchers around the world. And it already is being used by researchers around the world, but we want to do that even more so and create on-ramps for new kinds of research using this data. And so that's something we're going to be doing, where we provide data and allow researchers everywhere to try to, compete to develop algorithms to learn new things.
Nice. Yeah. Any other of the panelists want to?
Yeah, I can just mention, I put two things in the chat. One is an RO3 program that started, I guess, in 2019. It's still running. It's $100,000 per year for 2 years to get people who aren't already involved in Alzheimer's research, or more early stage investigators interested. So that's a great way to get started. And then there's another one that's probably more for more established investigators, but specifically around the idea of collaborating with these major infrastructure programs. So, and the last thing I would say is that if you have an interest and you're not sure who to talk to, you should absolutely feel free. I'm sure I can speak for everyone here to contact any of us, but also any program officer at the National Institute on Aging or NIH in general will be happy to help you get to the right person. And I will add that since everyone's so dramatically busy, if you write to any of us and we don't answer, please feel free to send a second poke or a third poke, because we're not doing it on purpose if we don't answer right away.
Wonderful. Dr. Wang, you're unmuted. Did you want to?
Yeah, I just want to say that the first is we have the genetics resource is really complicated, and that's because come from so many different cohorts and with different study designs, and that's the only way we can have enough statistical power. So that's why we have these websites, and we're really proud of the new websites, especially ADSP. We're going to continue to update it. If you have any questions, please send us. We'd like to hear what you think, and your feedback and questions really can help us to make our resource better as well. So I think these are best two ways to get to start accessing the resource.
Fantastic. Well, I don't see any more questions, and we're close to the end of our time. And I wanted to thank the GSA for asking me to moderate this. This is just fascinating stuff, and just so impressed. And it's such a model for other ways to approach very important health problems facing older people in a very, very deliberate, and I will have to also say very well-resourced way. So it's nice to see those resources being used in such a strategic and impactful way. I'm not certain if I'm supposed to turn it back to you, Jim, but if not, I'm turning it back to you. So.
Thank you very much. And I want to thank all of our participants in the audience. I want to thank you for your support of GSA. I want to especially thank the National Institute on Aging, the Alzheimer's Disease Centers for this wonderfully informative program this morning, today. And I'd also like to thank Dr. Krzyzewski for serving as a moderator for this panel. Do know that this program has been recorded and will be available very shortly on the GSA website, as well as the slides from all of the presentations. Thank you for your support of GSA, and look forward to future meetings. If you have suggestions, please also let us know that in the chat. With that, we'll close today's webinar and thank all participants. Have a good day.
Thank you.
Thank you.